All right, let's jump right in. Today, we're going to unravel what seems like a really simple question, but it's one that actually opens up the entire fascinating world of modern ALS research.
So what is it? How many genes cause ALS? You think there would be a straightforward answer, right? Well, it turns out this simple question is actually the start of a much deeper, more interesting story.
Yep, the answer is it's complicated. There isn't one single official number that everybody agrees on. And the reason for that is that different scientists are, in a way, counting different things. So to really get it, we need a better way to think about the problem.
So really, the best way to think about it is as a range. The final number you get depends entirely on what you're counting. Are you talking about genes that are absolutely confirmed to cause the disease? Or genes that just nudge the risk a little? Or maybe even genes that are just promising new leads?
So let's peel back the layers here and break down exactly why this question is so deceptively simple and what the answer reveals about the nature of ALS itself. This table really lays it out perfectly. You've got the established genes. We're talking over 40 of them. These are the confirmed heavy hitters. But then look what happens. The number grows when you add genes with smaller effects, and it just explodes into the hundreds when you start including all the potential candidates that are popping up in huge genetic studies.
Okay, so how do we make sense of all this? Well, researchers have this really useful mental model that I want to share with you. They organize all this complexity into what they call the 3 buckets of ALS genetics.
And here's our first bucket. These are the big ones, the high-impact genes. A mutation in one of these can all by itself cause ALS. And what's wild is that just 4 of them, C9orf72, SOD1, TARDBP, and FUSE, account for up to 70% of all inherited cases. Think about that. A tiny group of genes with a massive, massive impact.
Now, bucket number 2 helps us get a handle on the vast majority of ALS cases. This one is filled with risk and modifier genes. Now, individually, each one only has a tiny effect, but their effects can add up. It's like a series of small nudges that can push a person's risk up or down over a lifetime. This is a huge piece of the puzzle for understanding why the disease so often seems to appear out of the blue with no obvious family history.
And our third and final bucket? This one is all about discovery. It's overflowing with hundreds of candidate genes. Now, these aren't confirmed causes. Not yet. They're leads, uncovered by these massive genomic screens. They're basically signposts, pointing scientists toward new pathways to investigate and telling them where the next big breakthrough might be hiding.
Okay, this next part is absolutely crucial. We're about to pivot from just counting genes to understanding what all these different genes actually mean when you put them all together. This is where it all starts to click.
I want you to really let this sentence sink in because it is the key to the entire modern view of ALS. Genetically heterogeneous. That just means lots of different genes can cause it. But biologically convergent, that's the magic part. It means they all funnel down and attack the same core systems in the cell.
So here's the big takeaway. In the end, it almost doesn't matter which faulty gene a person has. It's like having dozens of different keys, right? They all look different, but somehow they all manage to unlock the very same door because they're all targeting the same few locks. So let's go on a little tour. Let's go inside the motor neuron and look at these core systems, these 4 key vulnerabilities that all those different genes seem to be attacking.
First up, we have RNA metabolism. The best way to think about this is as the neuron's internal supply chain. It's a super complex logistics network that's in charge of getting critical messages, the instructions from our DNA, from the cell's headquarters out to the front lines where the work actually gets done. So when genes that are part of the system, like TARDBP or FUSE, have a fault, the entire supply chain gets gummed up and starts to fail. And for a motor neuron, which has to send supplies down a pipeline that can be several feet long, that is a catastrophic failure. The whole system becomes unstable.
Next on our tour is protein quality control. This is literally the neuron's dedicated cleanup and recycling crew. Its whole job is to find damaged or misfolded proteins, get rid of them, and break them down before they can build up and cause a toxic mess.
And here's how it fails. Faulty genes basically tie the hands of the cleanup crew. The cell's recycling system just can't keep up with the mess. And so, these toxic, misfolded proteins start to clump together into what are called aggregates. These aggregates are one of the classic, tragic hallmarks of what we see in ALS.
Okay, now let's look at the physical infrastructure of the cell. Axonal transport is the neuron's internal highway system. It's constantly moving essential cargo,
things like mitochondria for energy, building blocks for repairs, up and down the incredible length of the neuron.
You have to remember, motor neurons are extreme long-haul cells, so any kind of traffic jam or slowdown on this internal highway is a disaster. The cargo just doesn't get where it needs to go. And the parts of the neuron that are the farthest away from the headquarters, like the connections to the muscles in a hand or a foot, they start to starve and fail first. This is exactly why symptoms often begin in one specific limb.
And finally, we have the neuron's emergency maintenance and defense system. This is the crew that protects the cell's most precious asset, its DNA blueprint, from all the damage and stress that builds up over a lifetime of really hard work.
See, motor neurons are these high-energy cells, and they just accumulate a ton of wear and tear. When the genes that are in charge of DNA repair are faulty, that damage builds up faster and faster. And this provides a direct, powerful link between the genetics of ALS and the single biggest risk factor for the disease, which is simply the process of aging itself.
Okay, let's bring this all home. Let's put it all together and build our final, much more complete and much more powerful answer to that question we started with. It's time to go from just a number to real biological meaning.
So this is it. This is the real answer. It's not just a number, it's a story. The story is this. There are dozens of genes involved in ALS, and what they tell us, what they all point to, is that ALS is a disease where the fundamental machinery of the motor neuron, its supply chain, its cleanup crew, its highway system, and its repair team is systematically breaking down. And that leaves us with one last, really powerful thought. If all these different genetic paths, all these different keys converge on just a few shared problems, it means researchers don't have to chase dozens of separate issues. Instead, they can focus on creating one master key, a therapy that fixes those core shared vulnerabilities. And that gives real hope for a treatment that could one day help everyone with ALS, no matter which gene started it all.